Okay, good. Our next speaker is Erika Fernandez. Erika has over 20 years of experience in global regulatory affairs across the US and international markets, and is currently the Associate Director of Global Regulatory Affairs at Gilead Sciences, focusing on early drug development in the inflammation therapeutic area. Maybe even more importantly, she is currently a doctoral student at USC in the Regulatory Science program, which I was student number 2 of about we won't talk how far long ago that was, Erika. It's a program. It's the only doctoral program in the world in regulatory science, and attracts basically students from all over the world. Today, Erika is going to talk about the FDA's program to phase out animal testing requirements.
All right, thank you so much, everyone. Let me share my screen. All right. Hello, everyone. Thanks so much for having me here today. And today I will discuss a little bit about innovative approach to drug development. In particular, the FDA proposal to move out or reducing animal testing and touches very superficial on the global regulatory impact. Just a quick disclaimer. And then moving on. So I just want to bring back just for just to color this conversation, the historic background, the animal studies started in 1938. So fast forward to today, a lot more in drug development. We know we have a lot more tools, but the requirements are still there. So in 62, they expanded the government through the efficacy amendments. It was expanded in 78, the GLP regulations that were very important to standardize protocols, 93 ICH guidance, and then back to our 21st century cures. So the FDA momentum towards the non-animal testing methods, what does that mean to us at this point, right? So the FDA has a predictive toxicology roadmap. They have been discussing that for quite a while. There's some working groups composed by academia, industry, and government. So to facilitate that conversation, what we can reduce or phase out. So in 2021, the NAMS reports were published. We will go through what the NAMS are and what is the importance in that discussion. In 2022, there are some statutory changes that makes testing option in regulatory submissions. And then finally, the paradigm shift is confirmed with the most recently activities from the FDA. So in April 2025, early this year, the FDA released the FDA roadmap. It's not a guidance. It's just like what the FDA is thinking about moving forward. This roadmap, it is explanation of the visions and desires of the FDA. A lot of things are not in place. So it is in baby steps. That's how I see. So the FDA has a 3-year plan to get all the animal studies, the phase outs, some databases, AI modeling validations, and they are moving towards to that goal. So just now, last week, maybe like December 2nd or December 3rd, the FDA released now is a draft guidance open for comments from the industry. So it's a guidance for monoclonal antibodies. The MABs were the first product class for streamlining for obvious reasons. And then the key changes, in my opinion, were very, very important was the elimination of the 6-month chronic tox study. Of course, they are a process to get that waiver, but we go through that. So the 3-month tox study becomes a default, so the baseline to move forward through phase 2. And there are some cases that no toxicology studies were required. I haven't seen those yet. The FDA brings weight of evidence requirements, which is a package. So it's a data package that will facilitate communication with the agency and evaluation if the 6-month chronic tox is not needed because the data it's already there. It's nothing that we would see in a 6-month animal study anyway, so there is no point. And there is also some special considerations when we are talking about pediatrics, that is reproductive toxicity in juvenile studies. So why monoclonal antibodies are the focus for the primary for the reduction? Because the predictability that we can use NAMs for those approaches. And also less cross reactivity, that is less information with animal studies, there's no point of repeating those studies in primates. And then cost limitation, and we could minimize that cost. And at the end of the day, it's a lot of animals that we can avoid using it. So moving to the streamlined approach, the 3-month toxicology study. So the draft guidance says that the 3 months would be sufficient to move on through phase 2 or through the drug development. In some cases, we can omit, but I haven't seen that yet. So that omission has to be supported with a strong package. So if there is no animal model, there is nothing that you can show safety at that point. So discussions with the agency. I've seen some discussions bringing AI, but I don't know if we are there yet. So as I said before, I have not seen a situation when the complete toxicology study was waived. And every step of the way, if we're planning to request a waiver from the agency, we should engage with them. We can't do anything just based on the next step, next logical steps will be very risky. So when the 6-month studies are not longer required, in my opinion, it's when you have a very clean 3-month study, you don't have cross-reactivity in human primates. Most of the time, it is available, clinical data is available. So phase 1 is finished. So you have PK, PD, clinical safety. So the compilation of this weight of evidence will make the 6-month study no longer required. And I have seen the situation before. So the FDA brings this weight of evidence framework to support IND submissions. As I mentioned before, it is a package where you include in vitro. So there are some labs there. For example, Charles River is working on NAMs, and we'll go through that situation, the in vitro studies. It could be precedent. It could be information with the same target compare with the other compound that would provide safety and efficacy insights. It could be in silico modeling using AI tools validated. You need to touch base with the FDA before using those tools. And then also, the human tissue data that demonstrate target engagement and off-target interactions. Omitting toxicology studies, it's for specific circumstances when ADA confounding is present, when animal studies are not feasible, and then non-binding species selected. There is no cross-reactivity. There's no point of moving forward. When we talk about special population, we need to be very careful here. The way of evidence approach will have to be tailored to that special population as well. And the consultation case by case to obtain the agreement with the agency. And as I mentioned before, we need to have adequate characterization to move forward in special population. But that's part of the December 5th guidance. So I talked a lot about NAMs, right? So NAMs, new approach methodologies. They are, I would say, modern tools that we can use today to modeling, to predict drug safety and efficacy and replace the animal studies. There are 2 main categories for them. They are in vitro human-derived systems and in silico and modeling. So the in vitro derived systems will be organoids. It could be also organ-on-chip device, liver-on-chip devices. So to put some color here, liver-on-chip device can be used for drug-drug interactions and then assess daily issues. So 87% of predictability you can get from those in vitro assays. So there is also a human immune organoids that can use for test, cytokines to release, and cell activations, very important in the inflammation and sometimes oncology. And when we talk about multi-organ human body on chip, that's something that I see in the future that probably phase one will be on chip. We could get dose, first in human dose. We could have effects, pharmacodynamics, of kinetics and dynamics, and we would not need to test in healthy volunteers. But that's in future. So in silico is pretty much we bring out the modeling, all the information that we have today, powered with AI. And we do have a lot of information today. That is a lot that needs to be validated, the coding, and the FDA is open to learn more about that. So there is partnerships. The FDA is planning to have this open database in toxicology where information can be shared and added to that database, allowing us to get more information and the weight of evidence gets stronger. So the more information, evidently, that the database would have and more confidence in the modeling, more is the probability of us moving forward without the animal studies. I see a lot of academic opportunities in this field. It could be in the NAM development, it could be the modeling. It's where the creativity is. So to bring back the research, to align the research, the in vitro, and provide specificity into those tests and support the predictability of the toxicology, it's very important. It's something that industry is seeking. Industry is looking for partnership. Industry is looking for labs that can provide support and service. And right now, it's taking a while. It's taking about like a year and a half to validate any NAMs that we try to work on. But once it's validated, you can use multiple assets. So there is a case study here when a NAM-based an in vitro study was accepted for IND submission. So there was data, as I mentioned, in vitro and in silico data were available and integrated, supported safety, and then it could move forward as IND enabling studies. Evidently, there are limitations and challenges on the NAMs. So I mentioned the validation it's complicated. It is a step that we need to do forward to those challenges. The reviewer variability that happens. We never know, right? Someone is a little bit more information or a different background that would impact our limitation too. And then the data sets, right? So it's a limited access to those data sets. So the FDA has a proposal to have this beautiful open database, but so far it's been a project. So moving to regulatory strategy. So a little bit how we integrate that to the IND development plan. So if we do have time in the early in the drug development, and the NAM approach can reduce tremendously costs. A phase two, toxicology studies to support a phase 2A can go around 4 million. So if we can have that work done in advance and then probably half or a quarter of the cost in validating those NAMs would be very beneficial. So the species appropriate for the model has to be relevant to provide exchange for the non-clinical data. And again, we need to early engage with the FDA. So as in my experience, there is being a lot of like a good exchange, positive exchange with the agency. There is no, I didn't feel like I was shut down in my first proposal, was this openness to hear more and to work with the industry. What do we have? And if we didn't have enough information at that point, they would provide us what exactly they need from us to move forward. So the current regulatory landscape for acceptance of the NAMs. So the discussion has to come with the agency. The waivers are granted case by case. The weight of evidence package are the gate, the gating to accept a waiver or not. It's important to articulate how the NAMs, how the new alternative methodologies, the in vitro, the in silico, your literature, your protocol will replace the animal studies. And if there is specific expectation, if you have a tissue chip stability, a cell characterization, and computational model verification, it has to be discussed with the agency. So what is the agency expecting from us? They are expecting quite a lot from us now because it is new for all of us. So when it's new, it causes some fear, adverse risk taking. So when we put together the weight of the evidence documentation, we're thinking about holistically. We're thinking about most likely safety, right? That point. So a justification why my 6-month chronic tox would be irrelevant. It's very important because you can back up with your in vitro study if you do have, or results from phase 1A or phase 1B to support your safety. And then you should have details like you are waiving the 6-month study, but you need to have plans if something comes up, how we're going to identify and to address any uncertainty, any, I would say, a limitation in your study that could impact safety as the drug development process continues. So I talked about the weight of evidence, what exactly they're looking for. They're looking for the target pharmacology, the biology information, and to review similar molecules, so the same class to benchmark. That has been very helpful because we can see how the competitors or how something out there it's more advanced in terms of research, and we can benchmark. There is therapeutic areas that they take more risks on oncology, virology. Sometimes the bar is a little bit lower because of the therapeutic area. But where you go to the dermatology, when you have sensitive or special patient population, the agency tend to be a little bit more conservative. And always interact with the agency. And then if we decided that the 3-month study, it's the only one that we need to move forward up to phase 2B, and we need to have a risk-based approach when we connect with the agency. So roadmap for success is how we build our strategy. We build a very strong weight of evidence. We map the uncertainties, what do we know and how we mitigate them, and we align with the FDA. And I believe if we have those 3 components and we go with a robust proposal to the agency, they will accept. So here's an overall global content. So it's not only the FDA that is looking to reducing and replace animal studies. The European Medicine Agency, EMA, they are moving towards, there is also a guideline posted a week and a half ago looking for, the 3 Rs and then how some animal tests can be replaced. Japan also and other countries in Asia are aligned. They are looking to in vitro. They are looking for Korea is ready to use blood organ on chips. They have the organoids there. The validation, as I mentioned before, is the critical path, but everyone is getting ready for that. And the harmonization, the global harmonization is important because at this point, there's some countries that just follow ICH. So if we don't have the flexibility to have a negotiation with the agency and just go by what the ICH asks, we won't be successful. So I see that is a great partnership with the major regions to allow us that flexibility and to think forward. That is an opportunity for us for reducing animal studies. And I have seen that. And that's what I had for today, and I'm open for questions.
Thank you very much. Any questions?
Hi, my name's Chelsea Bainey, and I am on one of the projects with Hydronovo, and we are working on a product that is a 505(b)(2) regulatory pathway, and we have submitted our pre-IND, and we worked really closely with Mike Jameson because the drug we are using as our reference API has a very strong historic safety profile. So in our pre-IND meeting, we tried very hard to push for a single species safety study. And we submitted this before, gosh, we submitted it about a year ago now. I'm losing track of time, but it was before some of the new publication of their animal model came out. So they denied our request for basically a single species, and we went back and even tried to ask again in a request for clarification, and then said, okay, if we do have to do it, what does our second species have to be? And we settled that the second species has to be a canine model, which we were reluctant to do. I'm curious if there's any opportunity for us to go back to the FDA. It doesn't seem like there's a great pathway other than turning our IND in. Without that, even though they requested it, and trying to create this body of evidence, as you said. The only other thing I have found is the opportunity to do a Type D meeting, but we'd probably finish our first species and then go and show, hey, it was so safe, we really want to try to argue again. Just thought you could maybe give us, if there's a new opportunity now that they've changed their guidance, or how you might approach this now. And I know Mike's been critical in advising us on this as well.
Do you have human data?
No.
No. Okay. So we were in almost the same situation. So we are going to do another Type D meeting. So we get requests for clarification. We got back, but we need to provide a stronger weight of evidence. I think the agency now is more flexible to. I think it's worth it to try another. If your timeline allows, I would push forward to get another Type D meeting and show that there is no need to testing animals in the second species.
Okay, and actually you bring up a point. While we don't have human data, we are discussing trying to do some in silico mining of that might help us of clinical data that might exist in charts. So maybe that combined with our first species would be a body of evidence that would be compelling enough.
I agree. I agree. Yeah.
Thank you.
You're welcome. Thank you for the question.
I think this is a good example of reusing an existing drug, and the sort of the holy grail is to get the existing company that holds the patent to allow you to cross-reference all of their human data, just as you're saying, Erika.
Yeah.
We're able to do that with two of our other projects, but in this project, we could not convince the original patent holders to allow us to do that. I think that would have made a difference in that argument because, as you said, you would have had, in this case, over 20 years of actual experience, different indication, but of that same drug at a lower dose. But again, could not get that from the company.
Oh, it's unfortunately because we had a similar situation, and then we had clinical data, and then we were allowed to use only one species.
Yeah.
Yeah.
Hi, Juan Tabos from the University of Pittsburgh. I appreciate this wasn't the focus of your talk, but what's coming down the road for devices or combination products that have a drug? How is the FDA going to start looking at them in terms of these models?
I don't know. I don't think there is anything going on, as we move forward. I think in devices, the in vitro, I think the NAMs are a great example how that because we need a validation. You need to characterize your liver on chip or your multi-organ on chip. I think there is opportunity in that. In the combination, I don't. I don't see how the animal studies would be impacted in a combination product because that will be part of your drug. So, when you have your drug development, so that's where we are trying to get a waiver. I see that when you have your final finished device, will be in your phase 2B, almost 3. That's when you put your device there. But at this point, my information is only in the early drug development.
Erika, is this your doctoral thesis topic?
No, it's not. My thesis, it will be in precision medicine, which is somewhat linked.
I was just going to say, if it was, we would continue to chase you down so that you'd have information for us.
Yeah, this has been my life for this last, I think, 8 months. We are back and forth with the agency.